Today we are going to talk about the pathogenesis, the topic we started earlier and tried to finish it. But don't run away. I understand your feeling, but I hope that it would not be boring. Rather, you get some kind of good information which can not only helpful to understand host microbe interaction in plants, but also in humans and animals as well. So it would not be bad idea to revise what we have already know about the pathogenesis. In the very early, we have talked about it, and we said that it is a series of events which could lead to the disease. The first stage of the pathogenesis is known as inoculation, where the host and pathogen come in contact with each other, and after that they recognize each other, and if the recognition is successful, then the pathogen tries to attach itself with the host cell. After that, pathogen tries to enter inside the host cell because all the nutrients present inside the host cell, and once it reaches there, it tries to establish itself. So establishment of the pathogen inside the host cell is known as infection, and at this point the pathogen share nutrients with the host, and as a result of this sharing, the host physiology is disturbed, which can be seen in the form of symptoms. So the period from inoculation to the symptom appearance is known as incubation period, and it can last for few days to more, and depend upon from pathogen to pathogen and from disease to disease, as well as the prevailing environmental conditions. But the pathogen at this point knows that if remain confined to a single cell or to a few cell, sooner than later the supply line is cut down and the nutrients are not available. So that portion when die, the pathogen will also die. So he tries to invade the neighboring cells, and this process is known as invasion, which we are going to discuss in next slide. The word invasion originated from the invade. It means when the pathogen invade the new cells in the near vicinity or near the infection site. So it varies from pathogen to pathogen. For example, in case of fungi, some of the fungi remain confined to the upper epidermis or subcuticle. So, like in case of apple scab, where the only the top of the tissue is infected, while the rest of the tissue remain healthy and hyphae never moves down. In another case, we can see the mycelium is present on the top of the leaves, and we can see a whitish silky growth of the mycelium. And while the fungi obtain its nutrient from the host cell with the help of ostomium, and these are root-like structure which obtain the nutrient from the cell, and then these nutrients are given back to the mycelium, which will grow, but never ever the mycelium enter inside the host cell. The third is the in the case where the pathogen establish itself in either in the intercellular spaces or the intracellular spaces, mean either between the cell or inside the cell. So the outcome is in the form of brown necrotic spots. But here the fungi remain confined inside the host tissue. So there is a diversity from subcuticular to surface, and then from inter to intracellular invasion in case of fungi. Unlike the fungi, the bacteria, there you can't see much diversity in bacterial invasion. Bacteria lives between the cell, not inside the cell, and they remain there. However, when the infection is too much and the cell loses its integrity, then these bacteria can invade to the intracellular spaces as well. Viruses are much different from fungi and the bacteria when we talk about their invasion. One thing is clear that the viruses never live on the surfaces, as well as viruses never live in intercellular spaces. Rather, they live inside the host cell, and they enter inside the host cell either through mechanical injury or with the help of insect vector. And once they are inside the cell, they move from cell to other cell by the openings which are known as plasmodesmata. These plasmodesmata openings are there for the exchange of material between the cells, but the virus also use these opening to move between the cells. Nematodes prefer to be in intercellular spaces, but it doesn't much matter because they can puncture the host cell with the help of a stylet, which is a spear-like structure. So, from with the help of stylet, they can easily obtain the nutrient from the host cell. Invariants are easily divided into two parts. One are known as local invariants and other are known as systemic invariants. If you look at your right side of your screen, you are clearly seeing a leaf on which there are brown necrotic spots and they are distributed all over the leaf, and even some of these spots merge with each other. But all these spots are present on this leaf only and they are not distributed in the different parts of the plant. So if they are confined locally, such lesions are known as local lesions, and they remain confined to that part of the plant unless some agency pick the pathogen from this leaf and distribute it to the other parts of the plant. Such invariants are known as local invariants. And if we move on to the left side of the screen, you clearly see a bacteria and then there is a xylem vessel. So water is moving upward. And if water is moving upward, the bacteria is also moving upward. And then it is distributed all over the plant. Such lesions are known as systemic lesions and which could lead to the complete death of the plant, like in case of wilting diseases. The next phase of the pathogenesis is colonization. In the colonization, we study both the growth and the reproduction of the pathogen. If you look at the screen, you can easily find a fungal hyphae present inside the host cell. And if the host cell provide the nutrients and the environmental condition remain suitable, sooner than later you can see that the whole host tissue is covered by the fungal mycelium. So it means that the fungi not only increase its size but changes its shape with the passage of time in the invaded tissue. The second thing is that fungi reproduce both asexually as well as sexually by the production of spores. But remember, the fungi during the growing season reproduce asexually because asexual reproduction provide it more chances to produce more spores, so it chances to invade the other plants as well as the new parts of the plant increase. But when the season is near to end, fungi produce sexual spores because through the sexual spores it produces new genetic recombinants which help its progeny to survive under new environmental condition and produce better offsprings. Unlike the fungi, bacteria don't increase its size. However, it increase its number, and we all know that bacteria reproduce asexually by the binary fion. And if the conditions are suitable, then a single bacterium can produce 1 million copies in a 24 hours. Viruses like the bacteria don't increase their size, increase their number, and by using the host ribosomal machinery. So, a single infected plant cell may have 1 lakh to 1 million virus particles. Unlike bacteria and the viruses, nematode change their size and shape. First there is egg, then juvenile, and then adult. And a single nematode female can lay 300 to 500 eggs, and there are 2 dozen of generation per crop season of the nematodes. So lot of inoculum is produced during the crop season by the nematodes. Once the process of colonization is completed, then the pathogen tries to reach out the healthy plant, and this can be done by itself, like fungi and bacteria with the help of flagella and nematodes with the help of muscle. But that movement is quite limited one. Most of the time, pathogen is taken away either by wind or by the rain or by the insects or the irrigation water or the agriculture tools or by the activity of the humans or even the animals or the infected plant material. So all these agencies help the pathogen to reach out to the healthy plant. But this movement is not restricted one. For example, in case of rust, the spores can be taken from one continent to another continent. So similar is the situation if you are importing an infected seed from another country, then you are also importing the pathogen of that country as well. So that movement is not the restricted one, and there are plenty of agencies which are helpful for the pathogen for dissemination. This picture is a brief account of all the events which we went through in the last few lectures, and the major event is the pathogenesis. So, pathogenesis is basically a series of events which could lead to the disease. The first stage of the pathogenesis is the inoculation. Inoculation is a process in which the host and the pathogen come in contact with each other. And they try to recognize each other, and if they successfully recognize each other, then the pathogen attaches itself with the host cell. After that, pathogen tries to enter inside the host cell because all the nutrients are present inside the host cell, and the secondly, if it is present inside the host, it can be protected against the environmental factors such as rain or the wind. So once it reach inside the host cell, it tries to establish itself. So the establishment of the pathogen inside the host cell is known as infection. So at this point, the pathogen not only establish inside the host cell but also start sharing the nutrients with the host. As a result of that, the host physiology is disturbed, and this disturbed physiology can be seen in the form of symptoms. So the time taken from the inoculation to the appearance of symptom is known as the incubation period. So the objective of the pathogen is achieved. Now it starts sharing the nutrient with the host, and it is also protected against the environmental factors. But pathogen is wise enough to know that this relationship can't prolong, because the sooner than later the supply line of the nutrient will be cut down. When the host cell die, the pathogen will also die. So before that, pathogen tries to invade the nearby cells, and if this invasion is successful, it ends into a colonization. In the colonization process, the pathogen increase both its size as well as its number. So new kind of spores and the cells or the eggs are produced, which are quite similar to the primary inoculum which initiated the disease. The primary inoculum ended into the primary infection. So this time, this inoculum is known as secondary inoculum, and this inoculum is taken by different kind of agencies like insect, wind, water, soil, human, seeds, and finally this secondary inoculum reach to the healthy parts of the same plant or the new healthy plants, and it causes an infection which is known as the secondary infection. So primary inoculum produces primary infection, and secondary inoculum produces secondary infection. But this whole process in a kind of circle, and circle has no beginning and no end. So if you start your cycle from the infection, or you start your cycle from the colonization or the inoculation, then still you are moving in a circle. The second important point is that in case of annual crops such as wheat or maize or barley or chickpea, which die after one growing season, if the pathogen want to remain viable, it should dormant when the crop is not present in the field. So the pathogen dormant for a whole season, either in the plant debris or in the seed or in the soil. So for the next season, when you grow the crop, this pathogen again attack, and again this cycle is repeated. If the dormancy period stretch over the winter, then it is known as overwinter, and if it stretch over the summer, then it is known as oversummer. So I hope at this point every one of you understand the pathogenesis in the plant microbe interaction. And interestingly, the pathogenesis process is same in case of human and the animal microbe interaction. If you understand the pathogenesis in plants, you can easily understand the pathogenesis in the human and the animals. So at the end, there is a small piece of advice to understand or remember the pathogenesis as a story, not as a lesson. Because if you remember it as a story, its shelf life is long, and if you remember it as a lesson to get good grades in exam, then its shelf life may be the exam day. So remember it as a story.